if anyone has a problem with the recording, you can log off now, but we're going to record this talk and post it on the GSI YouTube channel. And one last plug, we're going to be hosting the GSI Annual Research Symposium in November. Next week, October 18th is the due date for abstracts, so please submit your abstract. Without any further ado, I'd like to introduce Christopher Benner in this talk and take it away. Thank you, Chris. You can share your screen any moment.
Cool, thank you, Nelson. First of all, thanks for the organizers for inviting me. I really appreciate the opportunity to share my work with you guys. And I'll go ahead and jump right into it. Can you guys see my slides?
Yep, looks good.
Okay. Great. So today I'm going to talk about decoding regulatory DNA, and I'll cover a couple different topics, and I'll kind of lay that out in a second. I thought it would be worth it to start, even though the introduction maybe introduced me a little bit. I thought I'd kind of give a very quick rundown introduction to my laboratory and what we do. So really, as a scientist, I think my main goal is to really understand regulatory DNA. It's a very complicated system. Years ago, the triplet code for amino acid encoding, was discovered. It was very straightforward, made a lot of sense. Regulatory DNA is not the same thing, right? So it's a much bigger challenge. So, why do we want to study regulatory DNA and transcription regulation? Well, it's obviously very important in disease. So, I stole this on the right here. You can see this is a pathway diagram from a review trying to describe a coronavirus infection, right? The details here are not really that important, right? What is important is at the end, in these kinds of slides, you always see all these little arrows that point down to eventually where the nucleus is, and you have DNA and transcription factor motifs. And I think the main point is for a lot of these diseases, there's always a transcriptional response that it can be very important, for designing future therapies, or just simply understanding how our bodies and cells respond. So it's I don't really need to introduce this that much. Transcription is obviously a very important question when it comes to biology and human disease. Another part of this motivation is that as we study more about how our genomes function and how we vary amongst individuals, a lot of the variation that's been discovered that's associated with diseases has really been mapped to non-coding regions, right? So how to interpret those mutations and what they're actually doing in our cells has been a big question that's been very difficult to decode. So, as a laboratory, we really focus on transcription regulation. That's kind of our bread and butter, or main research interests. And so really trying to understand what the sequences are in the DNA that recruit factors, and then what are those factors that get recruited, and how do they kind of orchestrate the assembly of the transcription machinery to ultimately drive transcription. Now, of course, we try to study this kind of at an individual regulatory element level, but also these individual promoters or enhancers in our DNA don't function by themselves. They often function in the context of a regulatory element community or within full chromosomes, right? So it's also important to kind of understand that context and think about how regulatory elements communicate with one another, whether it's by looping in three-dimensional genomes, three-dimensional space within the nucleus, or by other mechanisms potentially that they're actually communicating. How the fundamental question of how an enhancer actually modulates the transcription of nearby genes. I mean, this is still, after a decade or two of really intensive work, we still have a pretty rudimentary idea of how that actually functions. And then as a laboratory, what do we do? What's our investigative strategy, right? So, we're definitely a genomics lab. So we try to do everything at a high throughput level as much as possible, for a variety of reasons. One, looking at mechanisms or patterns genome-wide are much more reliable and actionable than looking at single genes. So we're big on all the different techniques out there for doing genomics work. Really, we kind of focus on specialized transcription techniques and ChIP-seq and Hi-C for genome 3D structure. We're getting a little more into single cell methods, but since we typically don't do a lot of tissue work, those are a little less valuable than they are for other people. But certainly single cell technologies are a huge movement forward. And then, of course, the other thing we really do is try to develop software and analytical approaches to analyze this data and try to extract a mean out of it. And so, as mentioned at the beginning, probably the thing I'm best known for is working on Homer, which is a suite of programs for analyzing NGS data, and really, it's most commonly used for its DNA motif analysis and discovery algorithm. But as a laboratory, as we kind of grow and recruit new students and other researchers, we're trying to develop other tools. So I'm just giving you some examples down here. One is Mirlop, which is another kind of tool for DNA motif analysis that tries to relate motifs to kind of ranked lists of genes or relate them to their degree of regulation. So it's a little bit more of a sensitive approach to trying to figure out where motifs are. And another example here on the lower right is Metascape, which some of you might have used. This is kind of a collaborative effort between other people in the area to develop kind of a web application platform for doing like functional gene enrichment. So actually, like, for example, if you guys are doing Go enrichment, like on a list of genes, this is a great tool just to copy paste your list of genes and kind of hit one button and it will do a bunch of analysis for you, right? So that's been kind of a rewarding project to work on. It's actually gotten really popular, I guess for good reason, I'll say. But it's been a good project to work with. So and one other slide I thought to add, because this does come up, what does my laboratory really do, right, in terms of experimental systems? So we really study kind of innate immune signaling. That's one of our main systems. We like to study transcriptional regulation, mainly because the magnitudes of change are so strong, right? If you're going to study a system, you want to study a system where the gene increases 1.5-fold after stimulation, or do you want to study a system where it increases 1,000-fold, right? Well, 1,000-fold gives you a lot more latitude for investigation, right? Much stronger signal, much easier to investigate differences. So, one piece of advice if you're studying transcriptional regulation, go for the big signals if you can. And then I'm a bioinformatician who's trying to develop some genomics capacity. So we really rely on cell lines to do kind of NGS method development, things like that. And really some of the more interesting things we do are really through collaborations, like looking at infectious disease, opioid addiction, other types of things with collaborators to kind of get into more interesting biology where, requires more expertise to also understand those systems. So, really kind of as a setup here for today, I guess you could say I get asked this question a lot. As a genomic scientist who's done a lot of NGS analysis and looked at motifs and looked at regulatory DNA, what are the kind of the assays or the questions that are, or techniques that really give you the most benefit for analyzing your data? If I had to pick an assay to investigate my specific system, which assay should I choose, right? How should I analyze my data to get the most bang for my buck? What program should I use, et cetera? And if I sit back and think about it, and think about where my research is taking me and all the data I've looked at over time, I guess the punchline to this is that I actually would put in a plug for studying transcription initiation. I feel like that's one of the events that happens in regulatory biology that really focuses a lot of these different topics and mechanisms that you hear discussed, such as epigenetics, chromatin context, transcription factor, recruitment and activation. All that kind of gets integrated to recruit polymerase and actually start transcription. So, really, and I'm going to try to make a case for you today, really, that this is one of the signals that is, incredibly valuable to study to unravel a lot of the regulatory programs and mechanisms that we're interested in studying. So, the kind of the rough outline of what I'll talk about today is I'll try to introduce you to the experimental technique that we use to profile transcription initiation called csRNA-seq. And so I'll kind of describe this a little bit. To show you kind of how we use it and what we think it's good for. and show you a couple of examples of how this is applied, including some work we've been doing in the COVID-19 world. And then really at the end is kind of, I guess, my true passion, if you will, is using this type of data and kind of the analysis of regulatory motifs to try to figure out how our cis regulatory grammar works, or how to read our regulatory DNA. So using these techniques, we've got some interesting hints about how this might unfold. So I'd like to share that kind of unpublished work with you. Okay, so to get started here, I mentioned one of the signals that we think is the most interesting for attacking and understanding regulatory processes is studying transcription initiation. So let me kind of set the stage here a little bit for you. So what I'm showing you is a kind of a genome browser shot. This is the mouse genome, roughly representing maybe 30 kb of sequence near the SPY1 gene. So the SPY1 gene encodes a very key transcription factor, P1, that's involved in macrophage and B cell development and function. Now, this gene, the promoter region, is really, if you guys can see my mouse, kind of to the about 2/3 down the way, the region here, and at the very top, I'm showing a track that shows you the coverage of RNA-seq data. So this will highlight where the RNA is actually a stable RNA of a cell. So you see there's a lot of signal right where the exons are located in this gene, and then below I have a couple of common track other genomics data, in this case a TAC-seq, which maps the regions of open chromatin, so places where the nucleosomes are spread apart and you can other proteins can access the DNA. And then we have histone modification, so places where nucleosomes have been covalently modified with different modifications that roughly map out places where we have active regulatory elements and active gene transcription going on. So H3K27 acetylation is a very commonly used mark to map active promoters and enhancers, and the same thing with H3K4 dimethylation. So these are two different modifications that roughly kind of tend to overlap in the genome as it is. And so you'll notice that, where you have this, in this particular locus, there's an open chromatin region right at the promoter, and you can see there's a couple other open chromatin regions in other locations. And ideally, this region upstream is kind of a putative enhancer region. So when I talk about transcription initiation and its measurement, what do I mean, right? So, oops, here we go. So now I'm going to fill in some more data here, right? And so one of the techniques we've learned to leverage to actually measure transcription initiation is a modification of some of the other methods that have been proposed before, which is a simple strategy to isolate transcripts that have just initiated from the RNA polymerase. And so if I direct this is kind of a busy slide, I apologize, but if I direct your attention to the bottom here, it describes our csRNA-seq method. So csRNA-seq in this case stands for capped short RNA-seq. And the general principle is kind of highlighted in this kind of little cartoon on the lower left here. When RNA polymerase initially gets to a starting location, it'll quickly make an initial transcript. And really within about the first 20 to 30 base pairs, the 5' cap is already added to that initial nascent RNA. Now, eventually, the RNA polymerase usually kind of stalls there. Sometimes it will continue further. And eventually, if you have a true protein coding gene, the polymerase will continue much further down the DNA to actually transcribe that mRNA. So the idea behind this assay, though, is to essentially take the total RNA. So this is a very simple assay. You just take all the RNA in the cell, essentially size select it. And the idea is we'll isolate the short RNAs, so the ones that are about between 20 to 60 nucleotides in length, and then do some steps to select for RNAs that have a 5' cap. And the idea is that the RNAs that are short that have this 5' cap essentially come from places where RNA polymerase was initially transcribing, because as soon as it starts to elongate, that RNA gets too long, so it will end up not being in this set. And if they don't have a 5' cap, they usually are RNAs in other places, like, for example, microRNAs, pi RNAs, other types of short RNAs that are there. So anyway, as a result of this, performing this assay, which is not too hard to do, it's kind of like doing a ChIP-seq experiment, if just for perspective on the amount of effort and costs associated. we basically isolate these RNAs and sequence them. And when we map them back to the genome, we essentially can map where RNA polymerase was initiating. And so what you'll see up here is that you can see a signal from the csRNA-seq right at the 5' end of this of the SPY1 gene. So this would be essentially the promoter region of this gene. And just for those of you paying attention, or maybe not, hopefully everybody's paying attention. For those of you that are confused with the way this is set up, there's the signal on the top is kind of for the positive strand of initiation. So these will be signals kind of initiating, going from left to right. And then the signals on the bottom would be for the kind of reverse strand. So signals going from right to left. And so what you'll notice is that in addition to the promoter region where we have transcription initiation signals, we also see signals in other locations along this locus, and they tend to line up with where the other open chromatin regions are, and also where these histone modifications are. So this assay, not only does it kind of identify protein coding initiation sites where we normally think of the TSS or where transcription initiates, but it also identifies all the other locations in the genome where transcription is initiating that you may not appreciate, right? And usually in active enhancers, we actually see RNA polymerase recruitment and initiation in those regions as well. So as a technique, it very effectively maps where all the active initiation is essentially occurring in the cell. So, just one kind of point of clarification for this, this method is very similar to other types of methods that measure nascent transcription initiation. So the data it produces has a lot of similarity to GrowCap or ProCap. So these are methods developed by John Liss's lab for measuring true nascent transcription initiation. Very similar to Karen Edelman's methods like StartSeq. It also looks for short RNA sequencing, or a method that I was a part of the development of in Chris Glass's lab, 5' GrowSeq, another nascent initiation kind of technology. However, it is very different from other types of 5' TSS mapping. So many of you might have heard of CAGE or TSS-seq or 5' RNA-seq, or even 10x has a kit that will do single cell 5' RNA-seq. So if you look at the data from those types of methods, they tend to they do map start sites very well, but only really for protein coding mRNAs. The other types of transcripts that are much less stable, including enhancer RNAs and these other types of RNAs, tend to be underrepresented in those data sets. So if you really want to map like all the locations of initiation, you really have to kind of it's important to use one of these more nascent style methods like csRNA-seq to capture those signals. And just on the right here, just kind of show you a scatter plot comparison of the relative intensity that we measure, or from either csRNA-seq, which is trying to capture the nascent initiation sites, versus poly-A 5' RNA-seq. So this would be measuring the stable transcripts in a cell. Most of the mRNAs are kind of in this diagonal region. But there's a lot of transcripts that only show up with these other methods like csRNA-seq, and these are typically at enhancers for the most part, or putative enhancers, I should say. So what can we do with this type of data? It really is in many ways kind of a replacement data set for H3K27 acetylation ChIP-seq. So it really identifies a lot of the same regions. Histone acetylation and transcription initiation are pretty closely related. However, the csRNA-seq is a little more precise. Measuring initiation is a little more precise to the regulatory element, so it's a little more sensitive for doing a lot of these other tasks. We can obviously overlap lots of other types of data with this, like look at where variants are. We can do kind of pathway analysis with the places where things are regulated. In particular, we can do DNA motif analysis, which is something I'll be talking about more as the talk goes on. And then there's other kind of games we can play too, like we can look at how transcription positions change. So one thing I didn't really mention yet is that this assay is really single nucleotide resolution. We're mapping the initiation site. So if that initiation site changes, or the TSS initiates from a different nucleotide within a regulatory element, we can track that and kind of leverage that for discovery. And I'll show you another example as we kind of continue further along here. Okay. So just kind of want to show you an example of what this data looks like, kind of in, as an application of it. We, really to kind of show its effectiveness in studying kind of cell signaling and transcription responses, we applied csRNA-seq to one of our favorite systems, which is stimulating macrophages through the TLR4 receptor. So this elicits a very strong innate immune response in macrophages. It's sort of emulating macrophages coming into contact with gram-negative bacteria. So, basically they were mounting a defense response against that. And on the left here, so it's a little bit of a busy slide, so let me kind of walk you through it first real quick. On the top here is just kind of a schematic. We're taking bone marrow-derived macrophages from mice and treating them for a control or 1 hour with KLA, which is a component of LPS. And on the lower left here, really showing you what different types of data look like in this system. So on the top is kind of the mRNA. So this is the ACOD1 gene is a very strongly upregulated gene after TLR4 stimulation. Essentially, there's very little mRNA and then there's a bunch. If we look at what happens with csRNA-seq, you can see that there's a little bit of signal in different places within this gene where we can detect transcription initiation. But after we stimulate with KLA, we start to see big changes, and not just at the promoter of the gene, but also in other regulatory elements, in this case upstream. So these would be kind of putative enhancers or other distal regulatory elements in the region. And then for comparison, we also mapped H3K27 acetylation or ataxi. And I think the thing I want to kind of drag your attention to is this dynamic response when you have transcription. So, when you do this kind of signaling. So, in the case of acetylation, let's say to start, you can see a lot of these regions already have histone acetylation present, and that's even more true in the case of ataxi. So, if we map where open chromatin regions are, the open chromatin regions in many cases are pre-bound by existing transcription factors or already set up the chromatin in a very specific way. When we stimulate, it doesn't necessarily change these regions that much necessarily, right? So some regions obviously go through very large changes. However, in the case of monitoring what happens to transcription initiation, we usually see much more dynamic and sensitive changes as a result of the signaling. So you can see even in these regions where we already had open chromatin, we can see very dramatic changes in what the csRNA-seq signal is doing as a result. And to kind of drive that point home a little further, in the middle here, I'm showing you kind of what happens with the stable RNA in a cell. At 1 hour using traditional RNA-seq, we might see a couple hundred transcripts get upregulated as a result of that. If we monitor all the csRNA-seq locations, we can see, thousands, if not up to 10,000 sites significantly changing their levels. So this gives us a lot more power for discovery. So for example, we can take these 11,000 sites, apply DNA motif finding to them, and essentially without knowing ahead of time what's going on in the system, we can instantly find out that, we have very nice enrichment for motifs that match AP1 or the NF-kappa B transcription factors, which we know from years of research. These are some of the key transcription factor targets of the TLR4 signaling pathway, right? So, you can envision applying this type of experimental design to systems where you may not know what the transcriptional targets are. This can be kind of effective to try to figure out what they are. Just to kind of flex the muscle of this type of technology a little bit, since it only requires you to isolate total RNA and then do the assay, you can do things that are kind of difficult to do with other types of systems. So for example, we can do a very tight time course. So we took RAW cells, which are another macrophage cell line, stimulated them for various times and did csRNA-seq. And so on the right side here, you can see a very long time course where we can try to track the dynamics of transcription initiation over the course of this activation. In fact, we went back and did this at 2-minute intervals. And so you can actually see the initial wave. It takes about 10, 12 minutes for, let's say 8 to 12 minutes for transcription to really, or for the csRNA-seq signal to show up. I mean, I guess it might be pretentious of me to say that it's, that's when transcription starts, but our signal really starts to come up about 10 minutes after stimulation. So The other thing you'll notice that often happens is you can kind of see a little bit of a coordination between regulatory elements. So the promoter region is on the right side here, and these would be putative enhancers upstream. You can see that the promoter starts to really start to see expression about 10 minutes, 20 minutes, et cetera, and really ramp up. You can see the enhancer RNA at the same time, or the initiation at the enhancer kind of does sort of the same thing. They're kind of moving in a wave together where they're both initiating transcripts. And this could be very indicative of the communication between them. So anyway, this is kind of a cool, or at least in my opinion, a cool application of what we can do with this type of data. The other thing that's really cool, this is a little plug about it, you can apply it to pretty much any eukaryotic organism. So kind of as a proof of principle, the postdoc who developed this, Sasha Dudka, he went and grabbed samples of total RNA, because remember, it only takes total RNA, right? From a couple of different collaborators who studied various organisms across the tree of life, right? So the Nematostella is like a sea anemone, kind of a model organism for, early metazoan development, or early separation from humans in terms of metazoan development. Neurospora, so this would be a fungi, rice, so plants, and Capsospora, which is a protist in this case. And so, applying csRNA-seq to all these, we could effectively map where initiation sites are. And in many cases, just like in humans and metazoans, mammals, whatever, we can see evidence for transcription initiation events outside of the typical genic region. So these might be, let's say, enhancer RNAs in plants or fungi, et cetera. Now, I hesitate to say enhancer RNA. I mean, I realize that might be a loaded term. Let's call it non-coding transcription initiation in promoter distal parts of the genome. But anyway, it's a cool technology, and we've also tried to do this more in a pathogenic setting. So one of our collaborators here at UCSD was very interested in cocci, which is a fungus that is endemic to the southwest part of the US. That's common with kind of more rural agricultural settings, and it goes through two different life stages. So we tried to map initiation in their mycelia versus their spirule form, and we can even track what's going on with respect to, in many cases here, there was actually changes in the transcript isoform. So the start site of the promoter for this particular gene actually shifted as the fungi went through different life stages. So really, kind of in summary of this part, there's a lot of different kind of interesting applications that we could use this technique for, that if you're interested in studying where transcription initiation is or where active regulatory elements are, it'd be a powerful technique to investigate those things. Cool. So I'm going to kind of move into this. I set up kind of three parts, so I'll kind of move on to the second part here, which is we wanted to take this type of technology and apply it to a more, say, a timely problem, right? In this case, investigating the transcriptional responses in COVID-19 patients. So this is a collaboration with researchers here at UCSD, Michael Lamb, who's a pulmonary physician, and Nicole Kolfel, who set up these cohorts. And we went about to, this is really kind of a proof of principle, because we really hadn't used csRNA-seq to investigate patient blood before. So what the idea of the study was to take blood from patients in a semi-longitudinal experimental design, isolate the RNA from their blood, from full blood. So this would also include neutrophils, so not just PBMCs, but also neutrophils or other granulocytes, and process that for our csRNA-seq and RNA-seq to measure the transcriptional responses. And just kind of give you a quick summary of what the kind of the patient, where the patient samples were in this setting. We basically were able to recruit or get samples from several different classes of patients. So in this case, some that first of all, let me back up a second. All these samples came from patients that were pretty ill. So all of them were admitted to the ICU at some point. But some had a fast recovery. And on the bottom here, I'm showing you their what we call the modified Murray score, which essentially describes the severity of their respiratory illness. And if they had a Murray score greater than 2, this essentially means they were on a ventilator. So there's a set of patients that had a fast recovery, some that had a very prolonged recovery, and some that were ultimately fatal. So what do we do, right? So we basically took the blood samples from all these patients, and so there were some of them had a longitudinal data associated with them, or longitudinal samples. So and essentially took that RNA and either performed RNA-seq, so kind of a traditional RNA-seq approach, or mapped initiation sites using our csRNA-seq methodology. And so here I'm kind of showing you an example. This is what one of the patient profiles looks like in the genome browser. So this is the MX1 locus. This is a really key antiviral or interferon stimulated gene that is often induced in these settings. And so you can see that, you know, when this patient first presented at the hospital, they had very high levels of MX1 expression, which kind of receded over time, over the course of their hospital stay, until, in this case, this would be day 1, day 3, day 5, et cetera, and where DC is basically where they're getting discharged. And you can see that it worked pretty well. So we got signal, so the red signal is the csRNA-seq signal. It seems to be at the promoter region. And you can see that there's actually 2 separate regions where or even 3, where we're capturing initiation signals. And these regions line up pretty well with public data or previous published data from, in this case, open chromatin ATAC-seq or H3K27, which marks where potential enhancers are, and these can be stratified by cell types. Now, so one important caveat I'm just going to mention in case people are not thinking about it, you know, this data is a bulk assay, so we can't resolve this at the single cell level like some other studies or other techniques would. So for now, for this technique to work at that, to look at different cell populations, we would definitely have to sort out, which is possible. But you know, just one caveat, just, you know, make sure people are on the same page on that. So what do you do with data like this, right? I mentioned this is a prototype, so you know, part of it is discovering what we can do with it, right? You know, if we look across our full set of samples, you know, we have here I'm kind of obscuring which patients what and what time they were at, but we can essentially find signal for csRNA-seq at nearly 100,000 locations in the genome, and so we call these locations TSRs. That means transcription start site region. And I didn't really I apologize. I probably should include a slide that would kind of explain why we call it a transcription start site region. If you look at promoters, sometimes you can have multiple TSS that are closely spaced together. So, you know, to kind of disambiguate the terminology of a TSS versus a TSR. A TSR is essentially like a regulatory element, if that helps make more sense. So essentially what we did is we realized we can look at across all our samples, and we can essentially look at how different regulatory elements change over the course of all these different samples. And essentially cluster that to figure out which regulatory elements have similar patterns of transcription response across all these different samples versus which regulatory elements might be more different. And so, you know, to leverage kind of the fancy ways to visualize data and categorize it, we took our data, which is this is kind of the old way of showing a bunch of different data, like a heat map, where, you know, the red means it's high level of transcription activity versus blue would be low. We can recast this or remap this into 2 dimensions using a UMAP, which is what you commonly think of like in single cell data, right? And so on the right here is showing you a UMAP of these different regulatory elements. So each dot here represents a different regulatory element in the genome, and they're roughly organized by which regulatory elements are more similar to one another. So if we kind of compare the TSR B versus A, so these are 2 different, you know, regulatory regions. So B and A down here, you can see that they're roughly correlated in their levels. So each dot here is an individual patient, and this is log scale. So, you know, they're roughly correlated, so they kind of appear kind of near each other in this map. Whereas the C element here and B element are not so well correlated, so they kind of are appearing further away. So just to kind of orient you to how I'm trying to show the data, this will matter in a second. So we can take this UMAP, which kind of describes all of our different regulatory elements, and then we can start to think about, okay, so what do we know about these patients or other public data? How can we kind of try to interpret what happened in this experiment? So one thing we can do is we can look at each regulatory element and correlate it with how strong it was in patients that were more sick or less sick. So at the very beginning, I mentioned we can kind of score patients based on their respiratory distress, right? And so this is called it's captured in this entity called the modified Murray score in this case. And so what we realize is that when we look across our UMAP of different regulatory elements, some of these elements are much more highly correlated with severe lung injury than others, right? So in this UMAP, the elements on the far left side are essentially much more highly expressed in patients that have severe lung injury. We can take this kind of a step further. We can also say, okay, we don't have single cell data from this experiment. However, we can look at the public data, previously published data for ATAC-seq enrichment across different cell types, and relate, the ATAC-seq open chromatin that's found in monocytes or plasmablasts or neutrophils, and ask, how enriched are those open chromatin sites in the regulatory elements that we're showing on this map? So again, the regulatory elements that are typically associated with neutrophils, for example, are showing up on the left side of this UMAP. Or monocytes kind of appear in a couple different locations. Plasmablasts, or kind of say a B cell fraction, it shows up in this very small cluster down here. So the interpretation is that these regulatory elements in this region are very highly associated with open chromatin and plasmablasts. And if you look at the identities of these regulatory elements, a lot of them are, no surprise, for example, in the immunoglobulin heavy chain locus, right, which are exclusively expressed in B cells in this case. So we can kind of map the cell type specificity. And furthermore, we can kind of associate the activity profiles with the original patients. So if we consider the patients that we did a couple of control samples, they have very high expression in the regulatory elements on the right side of this graph, whereas the ones that had much more trouble recovering, the regulatory elements on the left side of this graph had much higher signal associated with those patients. Probably the more interesting thing we could do with this, though, is now that we've kind of segregated these different regulatory elements, we can actually go in and look at the DNA sequence associated with them and do motif finding to figure out what transcriptional regulators were likely driving their expression. And so this actually works pretty well. So if we go in and look at the different clusters, we can actually identify the enrichment of places where there's NF-kappa B motifs. So this would be kind of idea of pathways that might be driving kind of inflammation phenotype, right? Or STAT motifs, which might be indicative of IL-6 signaling, for example, right? Or interferon regulatory elements, such as the IRF factors that might be indicative of interferon signaling. So going through these diagrams or these different clusters, we can find different regulatory motifs that might indicate where certain pathways are actually occurring or being regulated within these maps. And just kind of taking that one step further, kind of what I was just describing, kind of laying it out a little more clearly, we can essentially find, we can look across different clusters of these different regulatory elements from looking at these patients and look for different motif enrichment across them. And these often correlate with very specific pathway functions. So essentially, we can identify the set of regulatory elements that seem to be responding, in this case, to NF-kappa B and or TNF alpha signaling. Or, like I just mentioned in the other slide too, a set of regulatory elements that really seem to be responding to interferon signaling, et cetera. And on the right side here is basically coloring the map where those different elements and different responses actually occurred. So one thing I think that's actually kind of interesting about this is that sometimes we see the same motifs appear in multiple clusters, right? So in particular, I'll highlight there was a cluster that was enriched for NF-kappa B that was associated with TNF alpha signaling. But there was another a cluster of regulatory elements that also had NF-kappa B motifs that were enriched. But this cluster was not associated with the TNF alpha, more to the canonical NF-kappa B pathway, but rather actually was enriched for lymphocyte cells. and was seen to be much more associated with TCR or essentially NF-kappa B activation associated with the adaptive immune system. And within this UMAP, you can see that the NF-kappa B regulatory elements, NF-kappa B enriched regulatory elements associated with the myeloid cells really segregated from where the NF-kappa B enriched regulatory elements that were associated with lymphoid cells in this case. So, I guess kind of the long story short, looking through this compendium of regulation data, you can start to pick apart the different response pathways that show up. One that was pretty interesting for us was to actually find a cluster of sites associated with glucocorticoid response elements. So that's in this guy on the near the bottom here. That it's pretty striking, because if you go back and look at the patients where these elements were highly active, these were all patients on steroid treatment to mitigate their disease severity. And if you go to, for those of you that are familiar with this kind of steroid or glucocorticoid treatments, like the FKBP5 is like a canonical target. If you go back and look at the data there, it basically lights up like a Christmas tree in these patients, right? Because they're all in these steroid drugs, and you can really see that evidence in their transcriptional responses. And then kind of finally, for just talking about some of this, exploring the pathways associated with COVID. Since we have these genomic locations or these regulatory elements where we see all these different activities, we can also relate it back to genetics data. So, the COVID-19 Human Genetics Initiative Consortium, they recently published in, I guess it was in June, where they did a kind of a big meta-analysis of trying to find variants associated with a more severe forms of COVID-19 in the population. So we can take those variants and then map them back to our same kind of UMAP here to show which regulatory elements they actually overlap with. And as a result, you can see like several of the variants are associated kind of where the STAT or ISRE elements are. So this would be kind of a classic antiviral response cluster. Some of them are associated with the NF-kappa B inflammation, et cetera. So it becomes kind of a powerful technique to kind of bridge where these variants associated with disease are associated potentially with transcriptional regulation. So we can also apply some level of statistics to this. So we use the RELY algorithm developed at the Cincinnati Children's Hospital, and looked for a statistical association between different classes of regulatory elements and the variants that were discovered by the consortium. So, for example, if we take the regulatory elements that are highly correlated with the lung injury scores, so that modified Murray score I mentioned, we do actually see an enrichment for variants that were identified in hospitalized versus non-hospitalized COVID-19 patients. So basically, we're seeing the variants associated with severe disease in this case are showing up in the regulatory elements that are active in a severely injured patient. So there's a connection there that it's kind of satisfying to see, right? This kind of business is a little tricky because they don't line up perfectly, and when you go look at where the variants are, they don't necessarily always line up with the transcription factor binding site or something the way you want them to. But there is a general association there that suggests that there's some signal and connection between the genetic state of the disease and the regulatory pathways or networks that are unfolding to combat it. So, just to have a little bit of a quick summary here, I think this kind of data can be very powerful to try to figure out what's going on with the gene regulation. One thing that you'll notice I'm kind of missing here is, I didn't say how, we cured COVID and, magically figured out how to, end the pandemic. And, I think, one of the reasons that we didn't, cure COVID is that, this disease really is pretty heterogeneous. So like if you, I kind of skipped over it, but if you look at the heat map where we profile all these different samples, there's a lot of heterogeneity in there. And if you take all the samples from people that had a prolonged recovery versus a short recovery versus a fatal recovery, each of those subsets has a lot of variation. And so probably for this to really be successful, to really nail that kind of question down in a way that's very satisfying, we probably have to greatly expand the number of patients, and which I don't think is necessarily a criticism of the methods being applied, but it might maybe more reflect also just the heterogeneity of these diseases and the difficulty it is to.
What we do in this experiment is we knock down NORAD, and then we're looking at the.
Oh, sorry. Oh, maybe that was, I don't know if that was a question or not, but okay.
That was, I had to mute a unmuted speaker.
Oh, okay, no worries. Okay, so in my last couple of minutes here, I kind of want to jump in. I'm not going to spend too long on this. I promise not to hold you guys past your end time, okay? But I think this is really cool, and that we can use these techniques to try to decode transcription factor or regulatory element grammar. So the idea here is that we have regulatory elements for transcription factors are recruited, they recruit polymerase, and we get transcription. Now, what are the sequences here that drive these different patterns of gene regulation? Now, I think just as a setup, we can identify transcription factors that seem to be important, but the reality is if you go to all these, tens of thousands of regulatory elements, the same transcription factors seem to show up all the time. But you still get specific patterns of regulation at the end of the day. And so, the general field has this idea that, the order, the spacing, the exact composition of these regulatory elements is important to actually fine tune how transcriptional responses unfold. Just to remind you in the way this data looks, if, here's just another locus where we collected some csRNA-seq data and relate it to some other properties. But the thing I want to draw your attention to is that the initiation data is really single nucleotide resolution. So here I'm showing you histograms that are essentially mapping how often each nucleus, each initiation site is actually used. And so you can see that there's actually very specific patterns of initiation that might occur in a given locus. So the idea is, well, can we start mining these patterns for additional information? So, one of the simplest things you can do is just calculate the nucleotide frequencies of all the sites in the genome where you have initiation. And what do you see? So this is actually what you get when you take all these initiation sites and look at the average ACGT content around the actual transcription start site. And so what you'll notice is there's a lot of interesting patterns that kind of evolved here, right? So right at the 0 point here is essentially where you can recapitulate the initiator sequence. So this is where the initiator elements where you typically initiate. So it's normally a CA. So that's reflected in the data. You notice there's this kind of surge in AT content upstream of the promoter or this transcription start site. So this would be kind of your canonical Tata box. And then, another thing that always impressed us is that, if you look downstream of these signals, you often get this 10 base pair fluctuation in nucleotide content, which kind of coincides with where you normally see a nucleosome position downstream of transcription. So the idea here being that, the actual, probably the rotational position of the nucleosome is probably pretty important for transcription initiation. So you can imagine if you have a nucleosome downstream, if you move it 5 base pairs, if you think about the helical nature of DNA, it's going to kind of flip it around to a different orientation, right? If you move it another 10 base pairs, it will kind of flip back up to kind of have a similar rotational position around the DNA. And so it's very likely that this kind of fluctuation down here might help manage that situation, although, not fully tested, right? This is a little bit of speculation on my part. Anyway, so there's a lot of interesting signals that you can see. More interestingly, though, we can also map where transcription factors are actually occurring. So, this is just kind of a simple plot to kind of give you a sense for what I'm going to be doing. We're going to take the location of where we find transcription factor motifs, and essentially add them up to get profiles so we can see exactly where they are relative to the transcription initiation site. So if we do that with different transcription factors, sometimes we have very interesting patterns, right? So for example, one of my favorites is the Nfy transcription factor. It's a very general constitutive transcription factor. We map its location across the genome relative to all the transcription start sites in the genome. You notice that it's not found very much downstream, but you can see this very nice helical distribution of it upstream. So about every 10 base pairs, you see a preferred location for the Nfy transcription factor. So again, this kind of presents a potential model here, right? Where the location of that transcription factor motif is very important, especially in this upstream region. if you move that motif about 5 base pairs in either direction, that really positions the motif on the other rotational surface of the DNA as of the double helix. So you can envision that if that motif is any position within 10 base pairs, it basically presents the same basic structural configuration. But if you put it 5 base pairs, it puts the motif on the factor that binds it on the other side of the DNA, let's say, in this case, right? So this kind of helps give you an idea of how these transcription factors might have important location preferences. And actually, if you map the different strand orientations of these motifs, you can see they're actually even out of phase with each other, which is kind of a cool illustration of that. You can see these kinds of relationships even in looking across different cell types. So if we map initiation sites in different cell types and look at where they are, they occur in different locations. And also in the context of signaling. So if we look at sites that are induced, in this case, we add KCl to neurons and look at where the CREB motifs, which is kind of the key binding sites that respond, you can see that we have very nice localization of these motifs relative to the start site. So it's very suggestive that the actual locations of these motifs might play critical roles in how transcription initiates. So the last little experiment I'm going to show you is what happens, how do we test this, right? Like, how do we perturb these factors and see what actually ends up happening? So you can envision a collection of regulatory elements. If we start knocking out a factor, how do these initiation sites maybe change as a result? So very basic biology in this case. So we started this by knocking out a transcription called NRF1. So this is a very constitutive, very common factor for a lot of different cells. So if we knock it down with siRNA and then repeat our csRNA-seq experiment, not only can we see places where the transcription changes, but we can actually see it where they shift, right? So this is a locus where there's actually several different regulatory elements. So here we're basically knocking out the NRF1 site, and we start losing some initiation sites, but we start to gain others or increase the transcription initiation rates at other sites. So one thing we can do for the analysis of this is we can take all these sites and basically rank them. And in this case, I'm ranking all the initiation sites on the y-axis. So the sites that kind of increase activity with siRNA are at the top, and the sites that decrease transcriptional activity after knockdown are at the bottom. And so on the axis here is where the transcription start site is, and all these little dots are showing you where NRF1 motifs are located after when we scan the genome and try to figure out their location. So something that was incredibly surprising to us, that's kind of reflected in what you see down below here, is that the NRF1 motifs were very dense upstream of the transcription start site, where they're required for activation. So basically, when we knock down NRF1, these sites all lose their expression. However, we saw a bunch of sites where the expression got increased, and in those cases, the NRF1 motif was actually located downstream of the transcription start site. And so we basically went through, analyzed this, looking at the correlation between motif presence and activation, and saw that, really the NRF1 site, the sites that are located upstream of transcription are, playing a strong role in maintaining activity, whereas it looks like the sites that are downstream are actually inhibiting transcription or blocking this activity. And so to kind of illustrate that kind of more in a different way, you can envision the NRF1 motif binding here and activating transcription. And if we get rid of it, this allows the RNA polymerase to load further upstream. So essentially, the NRF1 seems to have been blocking activation from upstream sites. It's almost like a buffering system, if you will, right? So to kind of pull that thread a little further, we tried another experiment. In this case, we added a dominant negative version of NRF1. So instead of depleting the factor and removing it from the system, we added a factor, a dominant negative version of it, to see what would happen as a result. And so surprisingly, or maybe not surprisingly, if we do the same type of analysis and sort all the transcription start sites based on whether or not they were activated or repressed with the dominant negative, we found that the ones that are repressed similarly are all, for the most part, upstream. But in this case, anything that was induced did not have NRF1 associated with it. So for us, this really kind of presents this model whereby the NRF1 motif, the factor is essentially, really sitting there and blocking the potential activation of other sites in this region. So as opposed to when you use siRNA to deplete the factor, the factor is gone, which allows polymerase to actually load. So really, I think kind of as a punchline for this, that it seems like that transcription factors do have the ability, even though they're like factors like NRF1 are considered very potent transcriptional activators. We can use them to, there does seem to be, convincing evidence at this point. And I'm going to skip some of the other things that I'm going to talk about because I realize we're at the end, that basically kind of help support this point. It looks like NRF1 and a lot of other factors actually can block transcription that's occurring upstream of them. So even though they're potent activators in their own right, there does seem to be some importance to the relative rules of where these transcription factors are placed along the DNA to dictate the location of transcription initiation sites. So I'm going to zip through this because we're already, I think, at 10 o'clock, or well, 10 o'clock for me. And really just kind of summarize this real quick, that, the csRNA-seq method is, really, we think it's very, using transcription initiation more than anything is a powerful technique to investigate transcriptional responses and deconvolve some of the mechanisms that are at play. And as I was just kind of showing you, using transcription initiation to kind of figure out what the regulatory logic has a lot of promise, right? And so we're trying to, also develop additional methods to help support that effort, and including some stuff I skipped over, actually mutating sites to see how the mutation of actual binding sites also kind of plays a similar role in either showing, supporting a mechanism of transcription factor blocking versus activation within these individual regulatory elements. Okay, so I realized I'm kind of getting to at the end, so I'll stop there with obviously what's usually the most important slide, which is really thanking the group and the people I've been working with. Really a special call out to Sasha Dudka in this case, on the upper left here. He's the postdoc who really developed the csRNA-seq method and really has helped driven a lot of this of the work that I showed here today. He's also kind of on the job market right now, so if you want to add a faculty member who's really interested in transcription, he would be a great grab. Also, special thanks to the Heinz Laboratory. So this is Sven Heinz is also a close collaborator who we worked a lot of these methods with, and the other collaborators for some of the other projects and funding sources as well. So with that, I'll take any questions. And sorry again, kind of get low on time here. Sorry about that.
Fantastic talk, Chris. That was really amazing. We have time for a couple questions. If you have a question, please unmute and ask. And I'll just start off with one quick question, Chris. I noticed on one of the slides with MX1, the csRNA-seq signal lasted longer than the RNA-seq signal as the patient progressed with time. Is that true? And do you have an explanation for why that might be the case? I think, that one. Good question. So these things are a little bit uncoupled. So the fact that the signal level here is the same height is maybe misleading. Okay. So you can see some RNA down here as well. The initiation signal and the, so if you did this at a global level, right? Like you could take the RNA-seq signal and the csRNA-seq and kind of correlate them. So they are very well correlated, but not always the same. And this gets back into the fact that transcription initiation is not the end-all result of transcription, right? So there's many other steps like to the control of elongation, telescripting, transcript maturation, splicing, et cetera, right? So we don't expect it to be perfect, right? But certainly they are pretty well correlated. One thing that is kind of interesting, you can take all the genes and look at which ones they are correlated with and ones that are less correlated. And that could be a little bit interesting. I think that's a thread we haven't pulled enough in terms of trying to classify genes that way, like which ones are mainly initiation driven versus, let's say, elongation or some of these other things. But I would say this is roughly typical that they're correlated, but yeah, you're right. Sometimes the initiation still might persist longer or precede it as well sometimes. So
Okay,
Thanks. Valentina?
Yeah, hi. So Chris, first of all, thanks. Fantastic seminar. It brought to my memories so many lab meetings where you were presenting wonderful data. So I have a couple of questions. The first one is about the promoter versus enhancer. So one cool thing about this technique is that you record transcriptional initiation in a lot of different locations, right? And it reminds me a little bit of one seminar where Karen Edelman had presented her model about how enhancers are not that different from promoters after all. So I wonder what you think about that based on the data that you have. And also if you have looked at any differences in your studies about the grammar of transcription factor binding sites and positioning, whether there are differences between the two cases.
Yeah, so to answer your question first, I am a huge believer that promoters and enhancers are more or less the same thing. You know, regulatory elements or active regulatory elements seem to not be very distinguishable when it comes to the sequences and their relative composition or even their epigenetics. I mean, there are some things that distinguish promoters and enhancers, like H3K4 trimethylation does seem to be much more common. Actually talking with Anna earlier today, you know, you tend to have splice donor sites just downstream of promoters, which kind of makes sense. They code genes usually, right? So, you know, the cause and effect there can be a little bit murky, although there's been a lot of data to suggest that, you know, a splice donor that, you know, recognizes the U1 SNRP complex might play critical roles in distinguishing or, you know, contributing to transcription elongation as a result. But by and large, it seems like promoters and enhancers are kind of one of the same type of regulatory element. And, you know, even the grammar stuff I mentioned before, like if I were to take all the enhancer elements and look at, you know, the distribution of different motifs and look at all the promoter elements, it's hard to tell a difference, right? I mean, there's GC content differences that can make people think they look different, but, you know, the reality is the relative positioning of the motifs and all that stuff, even Tata boxes, et cetera, are basically the same. So I would say there's not a huge difference. I would agree with Karen.
Thank you.
Are there any other questions from the audience? I have one more quick question for you, Chris. You had this ACOD1 gene or the LPS stimulation analysis, and you have one, yeah, this one here. In the one, if you just look at mRNA-seq, you only have 279 genes going up, and in the CSRNA-seq, you have 11,000. Does Homer perform differently if you feed it just the mRNA-seq genes, the 279 versus the 11,000?
Well, it will definitely perform different from the standpoint of like the confidence in the answer, because I mean, the more sequences you give it, it definitely does better, right? You know, it has more examples to learn from. You know, it's an empirical based algorithm, so the more data you give it, usually the better the outcome. Now, that being said, you know, LPS, if you look at promoter regions only, or, you know, mature transcript regions only that are induced with LPS, there's a decent number of NF-kappa B and AP1 binding motifs in those regions. So Homer probably, you know, I forget in this particular data set, but it probably picks up, you know, confident enrichment for NF-kappa B and AP1 in this setting. That being said, not all systems lend themselves to that. I feel like inflammatory signaling, you know, the data is so striking. It's, you know, the changes are so strong. It's kind of easy, let's say, right? Some of these other systems, though, where the signal is a lot more subtle, having those extra thousands of regulatory elements really makes a difference in identifying the signal. So I hope that answered your question. But
yeah.
Okay. Can I ask another very short question?
Yeah, go ahead.
So,
go ahead.
Sorry. So in the NRF1 story with the positioning before and after the transcriptional start site, do you see them being in different genes, or you think there may be cases where there is a switch of positioning when you go from repression to activation, and so the positioning of the transcription factor downstream of the transcription start site can be priming the gene to activation, if that makes sense?
Okay, yeah, I guess that's a hard question to answer, or I'm trying to figure out, I'm trying to dissect that question exactly. So generally speaking, the NRF1 motifs tend to be upstream of almost all the transcription factors, right? They tend to reside upstream of the transcription initiation site, in terms of the ones that are functionally active or seem to be functionally active. So I'm trying to think about your question exactly. This idea that the motif can be downstream of the initiation site. That initiation site is not always as clear until you get rid of the factor, right?
Yeah.
And so I think the problem with NRF1 in this case is it's sort of a constitutive factor, so it's usually there. So in a way, the sites, in this particular slide anyway, the sites that seem to be kind of upregulated that are further up, I'm not even sure physiologically how often this would occur, where these sites would actually get induced. So a piece of data that I didn't really show here is that, this kind of relationship, there seems to be some evidence for it, even with things like NF-kappa B, where you might have a factor where you're inducing it, or, you can also extend this maybe to developmental systems where you have master regulators or other transcription factors that are activated at certain times. But generally speaking, this kind of relationship seems to hold up, or at least the data we gathered generated so far, that if the factor binds downstream of the start site, it tends to repress things that were upstream, and then inspire kind of new initiation sites further downstream as a result. I guess when we've done this, we haven't seen a lot of evidence for a factor that stays bound but changes its mind, if that makes sense. At least not yet. And but maybe something to add to that just real quick. I don't know if this is getting too deep into it, but if you think about this experiment with the dominant negative NRF1, you might extend this to other. I mean, NRF1 is probably not a good example just because it's like this one constitutive family, but imagine things like BZIP factors like AP1. So AP1, there's like 30, I don't know, there's a lot of other AP1 BZIP transcription factors. Some of them don't have transcriptional activation domains, right? So if you overexpress one of those, or somehow translocate it to the nucleus, whatever, it might perform a similar function as what we see here, where it will block, basically, outcompete NRF1 or bind to new sites and block transcription that way. So that might be one kind of, I guess, extension of that idea.
Thank you.
Great.
Sorry, can I have?
Yeah, go ahead, Daphne.
Sorry.
Question.
Hi, Greg. Great presentation. Just to follow up on this, can it be that position-wise the transcription factor position is due to test-cycle effect?
I didn't really catch that. Sorry, it was muffled on my side. Sorry about that.
Sorry, I'll do this very quick. I'm wondering if you see this different position of the transcription factor and physiologically could be related to cell cycle effect. So at one point of the cell cycle, and NF1, for example, will be position 1 of the motif, while at a certain other point of the cell cycle is another motif.
Okay, I really apologize. I didn't really catch it.
Yeah,
Your question.
your audio is not coming through very clear. So
I'm sorry. Let me see if I can do this.
Can you hear me now?
yeah, better.
That sounded better. Yeah.
Is it better?
Oh yeah, much better. Yes.
Sorry. Very quick. It's the position effect of the transcription factor could be due to the cell cycle effect. So at one point you have the NRF1, at one point of the cell cycle, you have it on one position and then on the other. So could you discriminate for that, or
I would love to, except that, I think for this, and we talked about this in our lab actually, because, cell cycle is a super critical process, right? But we don't have data for cell cycle yet. Where we really went out and synchronized cells correctly. Like we talked to people about it. I don't know how many of you guys are cell cycle experts, but man, if you get a cell cycle expert talking about synchronizing cells, that's like, a good 3 hours that, they're just going to drone on about, right? So, I feel like we should get that data to understand how much it changes. Because, in theory, you're right. I mean, it seems like, in the cell cycle, there should be some cool differences, right? Where you have, these big important factors, especially constitutive factors changing because, you're totally, crunching up the DNA, you're doing all sorts of stuff. I mean, it's a great system to do. But, we kind of want to see exactly what happens, right? But, if there are big changes in the initiation sites, ideally, at least we would predict that hopefully that lines up with what's happening with those transcription factors. So I think it would be a very cool thing to study. But I would not speculate on whether or not we can kind of predict the cell cycle yet without kind of maybe seeing how that works. So
Thank you.
All right, thank you, Chris. I'm going to stop the recording.